Going. Okay.
Yeah, it's recording now.
Hi, everyone, and welcome to CHSS's next GBM speaker session. This week, we will be hearing from Dr. Dirk Dittmer. Dr. Dittmer was born in Berlin, Germany, where he got his BS/MS from the Free University in Berlin. He received his PhD from Princeton and completed postdocs at both Stanford School of Medicine and UC San Francisco. He worked as an assistant professor at the University of Oklahoma Health Sciences Center and is now a professor in the Department of Microbiology and Immunology here at UNC, where he also serves the roles of director of UNC Vironomics Core and team lead for Lineberger Comprehensive Cancer Center programs in virology and global oncology. Today, Dr. Dittmer will talk to us about his research on the interactions between cancer and HIV and AIDS. If y'all have any questions throughout the presentation, you're free to ask them in the chat and we'll get to them at the end. Dr. Dittmer, we are so excited to hear from you tonight.
Okay, there's that. Can anyone hear me?
Yeah,
Yes, we can hear you.
Can anyone see me?
No.
Ah,
Yeah, we can't see you.
why can you not see me? USB live camera is on. Oh, I know. Are you sharing my screen and that's why you can't see me? Is that an option? Oh, the camera is off. There's something odd here. Video. USB live camera. Okay.
There should be a little button in the bottom left corner that says start video.
Yeah, let me see. Can you not share your screen and then for a second? Now you can see me. Can you see me now?
Yes, we can see you now.
Wow. Hello, everyone. Can you hear me?
Yes, we can hear you.
Okay,
We did it.
then we're good.
All right, Jack, I think you can share the slides again.
Okay. Shall I start?
Yep, and do you have remote control of the slides?
Do I have control over the slides? How do I advance the slides?
You just click.
I just click. Ah, excellent. Cool. Hello, everyone. And thank you for, well, this one we'll ignore. Thank you for making time for me. It's really fun to get to meet you all. Would be more fun in person, but this will work. And in fact, I've been doing Zooms for quite a while now, even before COVID, because that's how you communicate with Africa. So let me tell you what I'm going to talk about in the next 20 or so minutes, which is how do you make a drug against cancer and how you do choose how to do that. I'm a member of the Lineberger Cancer Center, which is on the other side of campus, right after the dental school. And my group works on this disease called Kaposisarcoma, which is a cancer that develops in AIDS patients. If none of you are old enough, but in the 1989 around, those lesions that you see on the left, okay, hold on. Okay, anyone see this red dot here?
Yeah, we can see it.
Okay, so those lesions that you see on the left indicated people who had AIDS. The thing is, in sub-Saharan Africa, and Dar es Salaam is in Tanzania, so that's on sort of the right-hand side facing the Indian Ocean, and then if you go to the right, you hit India. In sub-Saharan Africa, this cancer had always been around, and it was a cancer of children, and it is caused by a virus called Kaposisarcoma-associated herpes virus. And it's still around. This is from 2019 when I was there, and that's hanging out there in the doctor's office to remind people to be on the lookout for these lesions. And then on the right, this is Zanzibar, where it's just gorgeous. Now, whenever you hear someone talk about drugs and science, they always have to put this disclosure up. I'm not sure if anyone told you that, but in biomedical science, we have to tell you everything and everyone who gave us money over the past 3 years. And so those are companies here and then the National Institute of Health. You have to have this disclaimer that says, whatever you're going to hear are absolutely only my opinions. They don't represent the University of North Carolina. No one is telling me what to do, what to say. And likewise, if I tell you something that's totally wrong, you can't sue the university. The other thing that I will talk about, of course, are experimental protocols and experimental drugs. And so therefore, we need to put a disclaimer saying, hey, I'm going to tell you some things that I think are really going to work, but we're nowhere near where they're going to work, right? This is the same as if someone tells you about all the SARS-CoV-2 vaccines that are in trials, but they have not been approved by the FDA. So for instance, I could discuss that I think that the Moderna vaccine is going to work in kids at the age of 14, and therefore all high school children should be immunized. But the trial hasn't been done and is only approved for 18 and older. So that's this disclaimer. This is the cancer, right? It's called Kaposisarcoma. It's whenever we look at a cancer, we want to know, well, where does it come from? It's an endothelial cell lineage cancer. Who has discovered? And if you discover a new one, you get to name it. So that's Moritz Kaposi, who at the time was in Vienna, and he was the head of the syphilis clinic because that was the disease everyone worked on at the time. Then, as I mentioned, Kaposisarcoma is a disease of AIDS patient, and then this husband and wife team had in UN discovered the virus in 1994. At the time, I was doing a postdoc at Stanford working on another herpes virus, and I decided this is what I'm going to spend my life on. And so I moved up the street to UC San Francisco, where they had just been able to grow the virus. And that's where this all emerged. And we're going to ignore the rest except these two things. One is the WHO has a commission that for every chemical and every virus decides whether or not it is potentially carcinogenic, and they meet and they vote, and you can look these up. They're called IARC monographs. And then this is our pathologist in Lilongwe, who I visited back in 2014, and he's looking at some of the sections. And so what UNC has done since 2014 is really engaged with a clinic in Malawi, which is now, we're going inland a little bit. It's above Zimbabwe, and below Zimbabwe is South Africa. And any given time, actually, UNC has about 100 people there at that clinic. We have our own hospital, obviously, but also sort of a little hotel and apartments. So if you want to go there, there are options for undergraduates. You just have to apply for them. And these are the data from UNC Chapel Hill. And you can see, for instance, that we see cases, right? NHL up here is lymphoma, is lymphoma. And then the next cancer we see in HIV positive patients is Kaposisarcoma. And they've been pretty much the same. And then the next thing you always look at is the pathology. This is a lung of a person who died of the disease. And you can see the lesions right there. And you can see more lesions there, and eventually the person expired. And trying to understand this kind of pathology is sort of the investigative part, right? This is, hold on. This is the part that House does. There's a patient, and we're trying to figure out what that patient is doing. And Kristen Tamburo in my lab was a grad student. And so this is the case, right? So all of clinical history starts with a case presentation. It's a 42-year-old Caucasian male. He was HIV positive in '93. He was doing fine. And in fact, he was so much doing fine that for various reasons, he just decided that he was cured and just continued medications 10 years later. And sure enough, 5 years later, he's at UNC in the ICU. And so he has all sorts of infections, and that's what these fancy names are, Clostridium difficile and so on. And we were trying to treat him, and it failed. And so one thing you do with viruses, you always look, what is the viral load, right? You heard that with SARS-CoV-2, and you can look for the mean viral load. This is a regular Kaposisarcoma. These are two other diseases found, and this is the patient, right? It's screaming high. And so that's when we go in and we just sequence everything that's there. And we find what the virus had. And of course, the virus had KSHV, the virus we were looking for. That's toxoplasma. Whoops. Somehow this is, let me take my point out. I think it's messing up with the way it works. It had, patient had toxoplasma and a whole bunch of other things. And so I want to move forward and be a little faster so you get sort of an idea. This is another case and another graduate student. This is from 2019, same idea. There's a person who failed HIV treatment and really progressed with his KS. This is an MRI or a CT scan, and these are the lesions. And this time we tried to treat the person with tocilizumab, which if you watch the news is one of the treatments they are trying. That we're trying, hold on, I can't read. Okay. We are trying the same medication that's used for SARS-CoV-2, which is sometimes also a cytokine storm, but it didn't work. And I'm going to skip this. It's just more data. So these are the kinds of data that are called diagnostic. This is what you get from the hospital, and you're trying to do on the spot, deciding what is the best treatment for the patient. And so my wife, Blossom Damania, she also has a lab at UNC. And I said, okay, that's all very good, but we really want to start and develop a new drug, right? And if you want to find a new drug, the holy grail is always a pill, oral bioavailable, because that's the only way you will ever be able to deploy a drug in sub-Saharan Africa. And it took 3 people to work on this. And we started at the beginning, right? There is this New England Journal of Medicine. If you have not heard about it, sort of every physician gets it. It's the most prestigious journal in medicine. And in Italy, they made this observation, right? I showed you two cases that we made. They had a couple more cases in transplant, and they found out if they take a transplant kidney patients and they switched the medication of what were they usually doing to this one called sirolimus or rapamycin, the cancer would disappear, right? And that's the smoking gun. That's in 2005. In 2006, we had figured out that there is a virus in monkeys, herpes virus Cymeri, that does something and gives you a phenotype very, very similar to what we were seeing in humans, in that it takes cells and transforms them in culture. And these remain dependent on a cytokine called IL-2, which is the target of rapamycin. And we did this thing by homology to say, okay, if there are T cells that are dependent on the cytokines and we have B cells that are dependent on cytokines, maybe the same drug will work. Now we're in 2007, 2 years later, we found two cell lines that you can grow in the laboratory. And the way you do it, you do the doubling time and we treat them with the drug and one worked, that's the blue one, and one didn't work. So now you have a positive control and a negative control, and you can look at the molecular mechanism, right? And so now we're going into cells, and we're going to measure again this human IL-6 cytokine, which we know is really high in patients. And we can show in the culture dish that if you give the blue drug, rapamycin, that biomarker goes down. So that's cool. Now we have a biomarker. Then comes the part I'm going to skip over. It's a whole bunch of trying to explain these things. And now we're going to spend 10 years from 2009 to 2019 to understand exactly what's going on. And we're still continuing to do that so that we know to the precise detail which amino acid is responsible for what, right? That if you think about SARS-CoV-2, we know there is a single amino acid, and if you change it, it will no longer or better bind the receptor. Same idea in cancer. We want to go down to a single amino acid that if we change it or if we observe it, we know the drug works. And you can see that here, this is the amino acid, serine 235 with drugs. They're all green, without drug, and you give rapamycin, and this modification disappears. And that's the molecular marker for the drug action. Then it goes into animals, right? 2007 to 2013, so that's 5 years. And this is just in mice. And we show again that the drug works in mice, and this is simply the area. So you get a lot of these charts, just ignore the numbers, but get a feel of that you move from culture to animals and how many years it takes. After animals starts the phase 1 clinical trial, right? And so this is now moving beyond UNC. There's a cooperative trial, that group that the NIH has set up, that involves people at Cornell, people at UCLA. They will enroll patients and they will treat them. In fact, they enrolled 7 patients, and that's on the left. ART regimen means what HIV medication they're on. Best response. Now I will try and get the pointer back. Best response is either a partial response, where sort of something happened, but not enough. Stable disease, meaning it doesn't get worse, but it doesn't get better either. And progression means the drug doesn't work at all. And so what you could see is that we had some responses. We stopped the disease in most of them, except in one which failed entirely. And then the fun part, not the important part, is we stopped it for up to 10 months. So Rapamycin is a pill. You take it once daily. And as long as you do that, your cancer will not progress for a year. So we just bought that person a year's worth of life, perhaps to allow them to live till there's a better therapy. And then we always measure drug levels. And then in 2019 comes the part where, in this case, the European guidelines change. And so now, from now on, whenever you develop Kaposisarcoma while getting a transplant, you will be moved to these inhibitors, and they have become what's called first-line therapy. So 10 years later, we actually made a difference. And that is incredibly gratifying to everyone who worked on the project. It means, however, that the person who started the project graduated a long time ago, and everyone who works on the last part has to only read the first part. And so right now in my lab, we're looking at already 5 or 6 molecules where we have the tissue culture stuff. And so in the 8 years, I can tell you if anything of that worked or if it all was all for nothing. And that is basic research. And that is my lab right now. All of them wearing masks and trying to find more molecules that can cure cancer. Happy to take question, and that's it.
Yeah, if you guys have questions, feel free to type them in the chat or just unmute yourself. But thank you so much, Dr. Dittmer, for that enlightening presentation. I'm going to be sending out the attendance form in the chat right now. So make sure you guys fill that out in order to get credit for attendance at tonight's meeting. Yeah, so if you guys have any questions, feel free to ask those now. But otherwise, thank you, Dr. Dittmer, and everyone for being here tonight. And we'll see you at our next GBM on Wednesday, March 10th. Have a good night, everyone.
All right, bye.
Thank you.
Mm-hmm.